Okay, so going into microbial genomics, right? So with our sequencing reads that are long as well as highly accurate, like we are actually you can actually run, a broad range of applications on for microbial genomics on hi-fi sequencing itself. So mainly, there are four primary ways that you can actually use PacBio hi-fi sequencing. So the first one, of course, is microbial whole genome sequencing. So, with hi-fi sequencing, you can actually get complete microbial genomes with ease and confidence. And for most microbes, right, uh, on our sequencer itself, you can actually get a closed genome as well as the accessory plasmid all assembled with just one touch using the default settings on our assembly pipeline. So you don't need to actually use short read to actually polish the genome itself as well. And if let's say you're doing viral sequencing, we do have some targeted approach that you can actually use as well. And for 16S, so of course for the 16S, like what I mentioned, we are doing full length 16S sequencing. So we can actually, go down to species and strain level so that you can actually look at your true diversity of your samples itself. And for hi-fi shotgun metagenome profiling, so for our hi-fi sequencing, right, you can actually get, you have better recalls and precision on the uh, on our hi-fi sequencing itself, and then you can also get richer functional information as well. And for finally, on the shotgun metagenome assembly, so with hi-fi sequencing, you can actually obtain many high-quality mags, and it's complete and closed genomes. And later on, I will show you more case studies as well. Our, running on hi-fi sequencing is actually more cost-effective as well for shotgun metagenome assembly compared to, other techniques. Okay. So I think there's a myth, right, that everyone, if I ask everyone on the floor, people will say, for PacBio, hi-fi sequencing, long read, is actually, it must be very expensive, correct, compared to, running it on Illumina or even MGI. So I think this is the perspective that we have from the market itself. But I think this particular statement is now outdated because, with this year, right, we actually launched, a few different workflows. For example, like our Kinex 16S workflow, as well as our Hi-Fi Plex prep kits, which actually, enabled new capabilities in the microbiome workflow itself. So right now, you can see that, for our Kinex 16S, uh, if you compare it with our if you compare with short read sequencing, if you're running on high throughput sequencing, you can actually achieve even cheaper price compared to what you are running on the NextSeq itself. And then as well as on the microbial whole genome sequencing, as well as the shotgun metagenome side. If you are using our newest Hi-Fi Plex prep kit, and on the Revio system itself, you should be able to get, even lower cost compared to what you're doing on the NextSeq. Okay. All right.
Um,
Hmm.
sorry, can you come back to the pricing?
Yep, sure.
This one is only for the sequencing, not include live prep and DNA extraction.
So it includes, so for example, like the Kinex, so for example, 16S.
Mm-hmm.
So how we calculate this price is based on the Kinex 16S kit itself, as well as the sequencing costs. Because we are able to actually multiplex a lot of samples in one single smart cell. So that's
Mm-hmm.
why we are able to actually, bring down the price. Mm. So it includes the library prep as well as the sequencing, but not the extraction part.
Okay, okay, thank you.
Okay, but just a disclaimer, so these prices are based on the US list price. So there might be some discrepancy in terms of the price in Vietnam itself, but this are references from the in the US. Okay.
Yeah, yeah, thank you.
All right. So, just to, going back to the platform itself. So for our Revio system, as I mentioned earlier, the Revio has four different stages, right? So you can actually run, different types of applications on the Revio itself. So on the different smart cells. So for example, you have run that is looking at you have an experiment that is looking at 16S as well as you want to look at their metagenomes as well, and then you do have some you know collaborators that are looking in viral and or microbial whole genome sequencing. You can actually run multiple different experiment in one single run on different smart cells on the Revio itself. And of course, if let's say you are doing other things like human as well, you can also replace some of the different smart cells with other plant and animal or human application also. So with the Revio system itself, with the increased chip density as well as the lesser time required, so we are able to increase the throughput. If let's say you are running shotgun metagenome or metagenome profiling by almost 10 times compared to the previous model that we have. So this is the reason, why the cost also goes down because, we are able to actually run more samples on one smart cell. Okay.
I have question, which mean
Yes.
that you have I can run some some application on single mast cell. I mean a single chip. It means that
Sorry, you were saying, yeah.
I can sequence 16S and metagenomic and microbiome whole genome sequencing just only one chip, one flow cell. Only
Uh, in this particular in this particular example, it's different application on different smart cells. But we do have customers that actually they try to run different application on one smart cell itself. Mm. So let's say you have some 16S and you have some metagenomes data as well. So we just need to make sure the library size are the same. Then you can actually run it on one single smart cell. But we usually recommend customers don't mix libraries together in one smart cell. Yeah.
yeah, thank you.